Okay, this is a review for the final to kind of give you some guidance. And let's just start with chapter 1. You are in anatomy and physiology, so I would expect you to know what those terms mean. Another test question on the test you'll find are these levels of organization. And so you should know that the cell is the level at which life begins. So cells are living, tissues are living, organs all the way up is living. Anything below the cell is non-living. So we learned about subatomic particles like protons, neutrons, and electrons that make up an atom, and that atoms bond together to form molecules. And we looked at macromolecules like protein, DNA, carbohydrates. And then we created organelles with those, like the mitochondria, the nucleus, the ER. And then that together works to help a cell live, the basic unit of life. So we've looked at different types of cells, cardiac tissue made of cardiac cells, muscle cells. We've looked at neurons as cells, glial cells like oligodendrocytes and Schwann cells. But those work together to form tissue, so muscle tissue, nervous tissue, adipose tissue, which is a type of connective tissue. And then we put those tissues together to make up organs, like the organs we learned about was the skin, the brain, the spinal cord, the skeletal, or like specific bones like the lung bone. And then we looked at organ systems like skeletal system, muscle system, nervous system, and those all make up a human. And so here's a graphic of that. We also learned about homeostasis and how that is really important to the body to maintain this internal environment. And there's sort of 3 parts to that: detecting any changes, relaying that to your control center, and then sending information to an effector to respond to those changes and correct them. And here's a graphic of that. Now, this is the common homeostatic mechanism, is negative feedback, where we are going in the opposite direction from the change in the environment. And so this can be body temperature, blood pressure, levels of everything in the blood pretty much. And then here's a graphic of negative feedback, comparing it to the thermostat in your house and the thermostat of your body. Positive feedback is uncommon, but we do see it with contractions of the uterus during childbirth, where there is a change that's in favor of the stimulus. And so here is an example of positive feedback, and here is the one we talked about in the body. So the body is made of 2 main portions, the axial and the appendicular portions, and we looked at the major body cavities. You'll need to know those for sure. So the cranial cavity, vertebral cavity, or spinal cavity, thoracic cavity, and abdominopelvic cavity. And then parts that help divide that: the diaphragm is that thin muscle that's between the thoracic and abdominal cavities. The mediastinum is between the lungs where the heart is. And then the abdominopelvic cavity can be separated into abdominal and pelvic cavities. And then we looked at this picture, and you should expect to know that for the test. We also looked at small cavities of the head: oral cavity, nasal cavity, orbital cavity, remember that's the eye, and middle ear cavities. We looked at serous membranes and how serous membranes fold on each other and make these 2 layers: the visceral layer, which actually touches the organ, and the parietal layer, which lines the wall of the cavity, and in between is the space where there's going to be serous fluid. So we have visceral and parietal pleura. Pleura means lungs. Visceral and parietal pericardium. Pericardium means around the heart. And visceral and parietal peritoneum. And these are the organs of the abdominopelvic. And we looked at this and saw that this blue membrane on top of the lung would be the visceral pleura, and the blue membrane lining the cavity of the lung would be the parietal pleura, and in between would be the pleural cavity. Same thing with the heart. The tissue sitting on top of the heart is the pericardial, parietal pericardium, the pericardial cavity between, right here, the dark space, and then outside of that, the parietal pericardium. The peritoneum, so we have the layer that's lining on the organ itself, that's the visceral. The layer lining the cavity, that's the parietal, and then you'll have the peritoneal cavity between. We talked about for anatomical position, that is where a person is standing up, facing forward, and their upper limbs are at their sides with their palms facing forward. And we learned that this man's left is actually over here, and this is his right. And these relative positions are important, so knowing all of these are on the test as well. And so we talked about right and left, proximal, distal, inferior, superior, medial, lateral, anterior, posterior, ventral, dorsal, lateral, medial, deep, superficial. And then we looked at the body planes, sagittal plane, transverse, and coronal, or frontal. So here is our transverse, here's our frontal plane, and then moving down the middle would be like a mid-sagittal. And then the anterior and posterior body regions, you should expect to label some of those. And that is all from Chapter 1 for this final. So there isn't any Chapter 2 or 3 on the test, so no chemistry, sorry about that, and no cell structure. That hurts my heart as a biologist, but Chapter 4, there's a little bit of Chapter 4 on the test when it comes to enzymes. We had just learned in Chapter 1 about metabolic reactions and homeostasis, maintenance, and we learned that these chemical reactions are sped up with the use of a protein, usually a protein called an enzyme. And what enzymes do is lower the activation energy so that the reaction can proceed quickly. And what happens is a specific structure called a substrate fits in the active site like a lock and key. And enzymes usually end in the suffix ase, like lipase breaks down lipids. So we saw these substrates, it could be one or multiple ones, fit together into the active site, and that forms the substrate, enzyme substrate complex. And what this does is lowers the active energy so that activation energy so that the chemical reaction can proceed and then a product is made. And we saw that oftentimes these enzymes work together to control a metabolic process in what's called a metabolic pathway. And so substrate 1 will fit in enzyme A, which will make a product, which is substrate 2. So substrate 2 is the product of enzyme A, but the substrate for enzyme B. And this just continues. And usually the end product will be an inhibitor to one or more than one enzyme to stop the process because you have enough product. And that's all on enzymes. So our Chapter 5 was about tissue, And we know that tissues are groups of cells that work together for a common function. And the study of that is histology. We learned that there are 4 major types of tissues in the body: epithelial, which lines and covers things; connective tissue, found all over the body; muscle tissue for movement through contraction; and nervous tissue to communicate with other cells and respond to the environment. And so here are those 4 major tissue types. And we also looked at types of intercellular junctions. Remember the tight junctions that fuse the cell membranes, desmosomes that kind of have little tiny buttons that connect them, and then gap junctions that are actually channels between cells. And so we see them here. This is the tight junction where it's really sealed the whole way through. You can see it's sealed here, but if liquid was in here, it wouldn't get through because here's the seal also. However, this is not sealed, so fluid could easily pass through this membrane because it's just being held together with a desmosome. And then channels allow material from one side, one cell to move, say, hormones quickly to another cell. We learned that while epithelial tissue does line and cover things, it also makes up glands. And it will always have a free surface on the outside, and it's connected to the basement membrane on the inside. And usually below that is going to be connective tissue. And we saw that epithelial tissue comes in different shapes and layers. And we can make all kinds of different names for those. And then we had some weird ones like the pseudostratified columnar epithelium, which looks stratified because the nuclei are at different levels, but really it's one layer of columnar epithelial tissue. And then transitional epithelial tissue has many layers, but it's stretchable. And so when the bladder expands, those tissues look thinner and elongated. So epithelial tissue does make up glands, and we had 2 types of glands: the endocrine glands that secrete fluid into the body, and then exocrine gland, which secretes fluid out of the body. We did not focus on unicellular or multicellular, but we did look at merocrine, apocrine, and holocrine, and there is a few questions about that on the test. So remember, merocrine, they're happy, they're merry because they are not breaking parts of their cells off. It's just the fluid through exocytosis. A piece of the cell is broken off and the whole cell breaks off for holocrine. And so here's visuals of that. So salivary glands, this would be some sweat glands, tears, apocrine, mammary glands, and then holocrine is like sebaceous glands. And then we looked at connective tissues, and they are the most abundant, and there is connective tissue proper, and then there's these specialized tissues, and they can be solid or fluid or somewhere in between, and they have many functions because they are so diverse in their structure. And so we talked about areolar tissue, adipose, and reticular tissue, dense, regular, dense, irregular, and elastic tissue. Those are your tissue propers. And then these specialized tissue, cartilage, bone, and blood. And so here are those, what they do, and where you'll find them. And then again, we looked at membranes. We've already talked and reviewed about serous membranes and the visceral and parietal and the fluid between that reduces friction. But we should also mention that mucous membranes line cavities that open to the outside, and they have goblet cells that secrete mucus. We also have cutaneous membranes. Remember, that is another name for skin, and it's part of your integumentary system. And then synovial membranes are found within joints. We saw synovial joints like in the knee. Muscle tissue, we have three types: skeletal, cardiac, and smooth. And skeletal muscle is striated. It's voluntary. It attaches to bone and it moves your body. And so you can see these long cylindrical shape cells with their multinucleated structure. Smooth muscle tissue, named smooth because it is not striated. They're more spindle shaped. They line hollow organs and blood vessels, and they are involuntary. While cardiac muscle is striated, which is why it's not called smooth, only found in the wall of the heart, and it has these weird branching cells. They are also involuntary, like smooth muscle, but they are neat because they have these intercalated discs. So here's the smooth muscle and the single nuclei, and they're kind of tapered. And here's the cardiac, and you can see these intercalated discs, and they're striated if you zoomed in. I guess I could make this. That does not even help us, does it? And then we looked at nervous tissue found in the brain and the spinal cord and the nerves that come off of the brain and spinal cord, like the 12 cranial nerves and the spinal nerves. We looked at neurons a bit in our last chapter, and the parts of a neuron, and then the neuroglia, such as the oligodendrocytes, Schwann cells, microglia, that sort of thing. And so here you see some glial cell nuclei, and then the main neuron with its soma, its dendrites, and its axon. So this would be a multipolar neuron. And so there is their description, function, and where you'll find them. All right, chapter 6 was the integumentary system. And so we focused on skin, right? The cutaneous membrane. And skin is made of two layers, the epidermis and the dermis. But we also talk about the subcutaneous layer, the hypodermis. And that's going to be areolar and adipose connective tissue that helps insulate the body and contains a lot of blood vessels, major ones. Of course, those break off into capillaries in the dermis that help feed the epidermis. Remember, the epidermis, like all epithelial tissue, do not have a blood supply. And so you can see this subcutaneous membrane below the skin, this adipose, and these blood vessels. And then we have this dermis of the skin, and then we have our epidermis of the skin. And we looked at these epidermis, dermis, and the hypodermis or subcutaneous membrane, and that there's a lot going on in skin. It's actually way more complex than you would think. There's a bunch of ducts for sweat glands and sebaceous glands associated with the hair follicle, and that dermal papillae that cause fingerprints, and then the hair papilla where vessels and nerves go into the hair follicle. There's all kinds of things going on in the skin. You do want to know the layers of the skin. BS, go cry, and don't forget stratum lucidum is only for thick skin. And so we're at our bottom layer. So if this is dermis, here's this dermal papilla, and this single layer is our stratum basale. And it is dividing quickly. It's well nourished because it's right here next to the blood vessels in the dermis. And then it creates stratum spinosum. And then spinosum's got some specific cells in it, but it changes to the granulosum, which is this part. And in granulosum, the cells are getting further and further away from the supply of blood, so they're dying and they're being keratinized. And then you have this lucidum layer, only found in thick skin, and then stratum corneum, which is our stratified squamous epithelium, and it is already dead and full of keratin and waterproofing for you. And so here again is a picture of that skin. And the accessory structures were hair follicles, nails, and these skin glands, and that's where we're focusing on. The final is sebaceous glands that make sebum. Remember, they're holocrine glands, and they're associated with hair follicles. Sweat glands are called sudoriferous glands, and there's eccrine, apocrine, and then ceruminous glands, which are earwax, and mammary glands, which are milk. Both of these are apocrine. And then, so here's a list. This would be a good table to look at. There's a couple questions on glands. Don't forget, the purpose of your skin is to protect you, allow you to sense information in the environment. It does get rid of some waste. Remember, this is where vitamin D is made so that you can make calcium and or absorb calcium and not have rickets. And it helps regulate body temperature by erecting the hair with the smooth muscle on the hair follicle, and also releasing sweat to reduce temperature. And that's all on chapter 6. So chapter 7 was the skeletal system, and we started looking at, of course, bones, and that they are living tissue, and many of them, and that we had axial and appendicular skeletons. We focused on bone shape, so expect a question on that. Long, short, flat, and irregular bones. Knowing examples of them. Let's see. So here are some bone shapes for you. The sesamoid bone being the patella, and then flat bones of the skull. And then you do need to know the parts of a long bone. So epiphysis and diaphysis. We do not talk about metaphysis. Articular cartilage. Remember, that's the hyaline cartilage that covers the epiphysis. This is the long shaft of the bone. These are the ends. The periosteum covers the bone. We have compact bone and spongy bone. There is a test question comparing those. Remember, spongy bone are what have trabeculae, compact have the osteons. In the medullary cavity or the chamber of the diaphysis, you'll find marrow. Speaking of marrow, there's red or yellow. Red is where hematopoiesis or blood formation occurs, and yellow marrow is where fat is stored, and the endosteum lines the space of that medullary cavity. And so here's a picture of the long bone. And so once again, osteocytes live in lacunae with canaliculi. And then again, the compact bone has osteons with a central canal going down the middle, and these rings around called lamella, where spongy bone have the trabeculae with lots of space between. And so this is showing compact bone versus spongy. And so there's some pictures of compact bone and an osteon with the central canal and these lamella going around. The major function of bones and what they do for us. We already talked about hematopoiesis, red and yellow marrow, and the epiphyseal plates are where the growth plates are. They are cartilage and they are ossified over time. If complete ossification occurs, it will no longer be called an epiphyseal plate. It is an epiphyseal line. Osteoclasts always break bone down and increase blood calcium, while osteoblasts take out calcium from the blood and build bone. And so a few things affect this building and growth of bone: the thyroid and the parathyroid hormone, sex hormones, and remember, physical stress actually working out makes your bone stronger. So this one is where muscles were attached and they were exercising. Much thicker of a bone than the one that did not. There are 206 bones normally in an adult skeleton, and so we have the axial skeleton and the appendicular skeleton, and don't need to know like how many of what, but you should know the bones of the skull, the hyoid, the vertebral column, the thoracic cage, the pectoral girdle, upper lower limbs, and pelvic girdle. And so here are major bones of the body: the hyoid, the skull. Of course, you can pause these. I should go a little slower so you can pause them and study them if you need to, or if you've printed out all your fill in the blank study guides, or fill in the blank PowerPoints, I would be like tagging these things. So there's the vertebral column and the coccyx and the sacrum, and then the pectoral girdle showing the clavicle and the scapulae and how they bind to the manubrium of the sternum and the proximal end of our humerus. And then you see the radius and ulna, the carpals, metacarpals, and phalanges. The lower limb, the femur, patella, fibula, tibula, tarsals, metatarsals, and phalanges there. The calcaneus bone, the talus bone, and the foot. And that is all on bones. I say that's all, but it's a lot. So make sure you can identify those bones of the body. A couple questions on that. All right, chapter chapter 8 is about joints. And remember, a joint is actually an articulation where two bones come together. And we classified joints based on their structure as being fibrous, cartilaginous, or synovial. And then we also labeled them on the amount of movement. Synarthrotic, meaning they cannot move. Amphiarthrotic, meaning kind of move, can move a little bit. And then diarthrotic, which is freely movable. So fibrous joints are held together by fiber tissues, connective tissues. And these are usually found in bones of close contact. So we have syndesmosis, sutures, and gomphosis. That's hard to say in the plural. So syndesmosis are found in these sheets. And so they are amphiarthrotic. You can flex it a little bit or twist, and this is found between the tibia and the fibula, as seen in these spaces here. Fibrous joints include sutures as well. We saw that in the flat bones of the skull, where we labeled the coronal suture and such. The gomphosis is like our teeth, and we labeled the parts of a gomphosis suture, so expect to do that again. Synovial joints are all diarthrotic, so freely moving, and have a very complex structure. They have all these bursae and synovial fluids and menisci. We also looked at types of movement of joints, so flexion and extension, hyperextension, lateral flexion, abduction, adduction. These are all test questions. And then dorsiflexion, plantar flexion, circumduction, rotation, medial and lateral, supination and pronation, inversion and eversion of the plantar, protraction and retraction, and then elevation and depression. And then examples of synovial joints, these complex joints are the shoulder, elbow, hip, and knee. All right, in chapter 9, we studied the muscular system, and we reviewed that there are 3 types of muscle tissues, and skeletal, cardiac, and smooth. And we did focus on skeletal muscles, and let's see. So we looked at the parts of a muscle, the fascia, the tendons, and then the epimysium, perimysium, and the endomysium, the connective tissue that kind of compacts these muscles. And so you can see that the tendons connect muscle to bone. And then you'll need to know this picture for your test for sure. And remember that these filaments are actin and myosin that make up the myofibrils. Myofibrils are the organelles within the cell called a muscle fiber. And you can see the nucleus there of the muscle fiber. Here's that sarcoplasmic reticulum that holds the calcium for muscle contraction. And then a group of these cells is called a fascicle. The muscle cell itself is wrapped in endomysium. The fascicle is perimysium, and then epimysium around the group of fascicles, the organ muscle. And then you'll have fascia on top of that. Remember, skeletal muscle is multinucleated. It has the sarcolemma. Remember, sarco can mean muscle, so that's the cell membrane of the muscle. The sarcoplasm is the cytoplasm inside of a muscle fiber. And we talked about the thick myosin and the thin actin and the sliding filament model, which we'll look at again. The sarcoplasmic reticulum stores calcium. The T-tubules allows the action potential to move down deep into the cell. And then we have these units of muscle called sarcomeres. And they make these patterns like the Z lines in the H zone and the I band. And when these contract, these Z lines come together and our A band increases. So you can kind of see that here. Remember, the thick filaments have these myosin heads that are going to grab onto the troponin of the thin filaments on the binding sites. Troponin and tropomyosin move to expose those active sites on the actin for the myosin heads. And so you can see the troponin here and the tropomyosin actually blocking the binding sites. When calcium's present, this troponin changes shape, which therefore changes the shape of the tropomyosin and exposes those binding sites for the myosin heads. Remember that to let go, you actually need ATP. And how does all this happen? Well, a neuron tells the muscle to contract, and this is at a neuromuscular junction. And so what happens here is the motor neuron has these vesicles filled with neurotransmitter, and those release that neurotransmitter into the synaptic cleft from those synaptic vesicles, and those bind to the motor end plate. And so then it will cause the active potential to occur and run down the sarcolemma into a T-tubule to contract these sarcomeres. And so here you can see the synaptic vesicles releasing their acetylcholine, and that is the major neurotransmitter for skeletal muscle. So the Z lines move closer, the H and I bands are narrow, and these actin and myosin filaments do not change shape, but they slide past each other, and that's how we contract our muscle. And we form these cross bridges. Remember, not only do you need ATP to let go and to release, but you're going to need acetylcholinesterase, which is the enzyme that breaks down the acetylcholine so that no more action potentials travel down the sarcolemma. And then calcium pumps move calcium back in the sarcoplasmic reticulum so that the binding sites can return to being covered. All of this has to meet threshold of an action potential for a fiber to contract. And there are types of contractions, both isotonic, which are moving where the muscle actually moves. You have concentric, where you're lifting an object by shortening the muscle, and eccentric, where you're lengthening your contraction and usually letting something go. Isometric would be, yoga stances or holding a weight in the same position. You're still contracting, but you're not moving the muscle and not changing its size. We looked at origin and insertion of muscles on bones, and we looked at major skeletal muscles, both anteriorly and posteriorly. Be prepared to name major muscles, compare actin and myosin, know the sarcomere and the bands and lines, and the parts of the skeletal muscle. And so these are muscles of the face. We didn't study all of these, but many of these we did. More muscles to help you chew. And so we labeled those. And these are muscles of the abs and the arms, and the chest, trapezius, shoulder muscles. Don't need that at all. External obliques, rectus abdominis. Let's see, we did not study pelvic floor, so don't worry about that. Gluteus maximus, abductors and adductors, gluteus medius, bicep femoris, semitendinosus, semimembrananeous, rectus femoris, laterus, vastus lateralis and medialis member on either side of the rectus femoris. Don't need that. Tibialis anterior, gastrocnemius, soleus, tibialis posterior. Don't need the cross sections, and don't need the lifespan on that. And that is chapter on muscle. All right, chapter 10. We are getting close, guys. I'm excited for you. Chapter 10, we studied the nervous system. We talked about sensory input, how we make decisions on that with interneurons and the central nervous system, and motor output, where the efferent pathway leads to muscles and glands. We talked about neurons being the nerve cells that do conduct these electrical impulses and use neurotransmitters to communicate with other cells, and the neuroglia helps protect, support, and insulate these neurons. And so we saw sensory information coming into the brain and spinal cord, and motor output going to the effector cells. We learned the brain and the spinal cord make up the central nervous system, while everything else is peripheral. And so we looked at the sensory division and the motor division, and further discussed how the motor division could be autonomic, automatically happen, or somatic, where it's skeletal muscle and purposeful movement, voluntary movement. And so we looked at those divisions, and here's a graphic of it. We said that the main structures of a neuron are the cell body, or the soma, the dendrites, and the axon. And we said that the axon hillock is where the cell body rises to the axon, and then at the end of the axon is the axon terminal, where you're going to find the synaptic knob and those neurotransmitters. We've talked about Schwann cells and how they insulate the axon, forming myelin, which is a mixture of fat and protein, and this helps speed up the action potential down that axon. And the nodes of Ranvier are the gaps in the myelin sheath where saltatory conduction occurs. And so we looked at a common neuron and labeled its parts, and looked at the myelin sheath around the axons. But we said that not all axons are myelinated. So myelinated axons are what make up white matter, and in the peripheral nervous system that is done so by Schwann cells, but in the central nervous system are the oligodendrocytes. Unmyelinated axons are going to be gray matter, and they are slower to transduct information. And we looked at this graphic. And then we looked closer at the neuroglia, and you'll need to know these for the test: the astrocytes, oligodendrocytes, microglia, and the ependymal cells are in the central nervous system, and we labeled those in this graphic. And then the peripheral nervous system just has the Schwann cells, which produce the myelin sheath around the axons, and the satellite cells that nourish and balance the ion concentrations of the neurons. And so here are the types of neuroglia, their characteristics and functions. We talked about action potentials more and how the cell membrane is polarized, meaning it is negatively charged on the inside and positively charged on the outside. And this is mainly due to the sodium-potassium pump and this uneven concentration of these ions. So at rest, the membrane is charged at about negative 70 millivolts. And if you reach threshold, you'll fire an action potential where all your gates open up, the sodium gates depolarizing the membrane, and we close those sodium gates and open all the potassium gates, and we repolarize, maybe hyperpolarize our membrane, but return back to resting potential. In chapter 11, we talked about major parts of the brain, and we started with reminding ourselves what the central nervous system is and learned about these meninges, the dura mater, the arachnoid mater, and the pia mater from superficial to deep. And that mater means mother. And so you have that tough mother on the outside, and the pia mater is that very delicate meninge that is directly on the brain and the spinal cord. And we looked at how the parts of the brain, and here are those meninges found in both the brain and the spinal cord. We see the cerebrum, we see, the cerebrum, the cerebellums, this curly one in the back. We can see the scalp and the gyri and the sulci and the bones. Here's a suture. And so we also looked at the meninges of the spinal cord, and we made a note that the gray matter is on the outside of the brain with white matter on the inside, where in the spinal cord, the gray matter's on the inside and the white matter's on the outside, so they flip. We also looked at the 4 ventricles, and these are just cavities within the brain and spinal cord where cerebral fluid can flow. And we looked at our first 2 ventricles called the lateral ventricles, and those lead to the 3rd ventricle in the midbrain and the 4th ventricle in the brain stem. That the cerebral spinal fluid is secreted by the choroid plexus and then absorbed by the arachnoid granulations. And so we labeled some parts here. We looked at the major portions of the adult brain, the cerebrum, the diencephalon, the cerebellum, and the brainstem, and labeled those parts. And we talked about gyri and sulci and the major fissures. The corpus callosum connects the 2 cerebral hemispheres of the cerebrum, and really the different lobes of the cerebrum. Again, gray matter on the outside of the cerebrum, white matter on the inside. And then these different regions like Broca's region and Wernicke's, and how this is the visual. Remember, we just learned about eyes in this last unit, and how those neurons cross and then finally meet back here. We don't worry about basal nuclei or Parkinson's disease. Let me look at my list of things real quick. And that's pretty much it on that chapter. I'm not going to review the final chapter 12 because that's new information. There will be 10 questions, and it's a really short lecture, but this will give you this is a study guide for you. So I know this is going quick, but the semester's at an end, and so please finish your final on time. I've enjoyed teaching you. I hope you've learned a lot. I hate that it's so fast. I really wish you could have been in person, way more fun, and the learning is so much better. But this is I understand escalating and helping you get to your goals faster. So I hope you enjoy the rest of your summer. And if you're not taking class, maybe I'll see you in Anatomy 2 in the fall.